morning, guys. Today we are going to learn about the cell membrane. Well, we're going to review cell membrane structure real quick, and then we're going to talk about transport and how molecules actually move from outside the cell to the inside of the cell, or inside of the cell to outside of the cell. So first, let's start with this membrane. If you're at home, this diagram is in Google Classroom. You can print it out. It's linked to the post for today. So you were asked to label it. So let's go through what each part is. One of these molecules right here with the head and the tail, that is a phospholipid. And these proteins right here that span the entire membrane, this is another one here. These are integral proteins. Specifically with this opening, we would call this one a channel protein. This one is also an integral. We also see cholesterol here. This structure right here would be our cholesterol. And let's see what else we need to label here. This would be a peripheral protein. These pieces are carbohydrates. And carbohydrates, when attached to a protein, and this diagram does look different than the one that we had yesterday, but this is representing a protein, and it has a carbohydrate attached to it, so this whole thing would be called a glycoprotein. Remember, that is used like an ID tag. It's used to identify the cell. Here we see a carbohydrate that's attached to a lipid. This would be a glycolipid. These spaghetti-like structures that we see coming off of the membrane and all throughout inside of the cell, those are the cytoskeleton. That gives the cell its structure. It does attach to the membrane to help shift the membrane and change the shape of the cell as the environment dictates. Then let's go ahead and label the areas inside. We know that this area here is nonpolar and hydrophobic. We know that this area here where the phospholipid heads is polar and hydrophilic. All right, so this is just a quick review. We're going to look today and find out how some molecules can move through the phospholipid bilayer and how some move through proteins, and when they need energy and when they don't. We're going to learn several different methods of cell transport. So let's look at cell transport. And just for a second, I want you to think about how molecules can get in and out of the cell. And just write down any ways that you think molecules can get in and out of the cell. And once you've thought through that, then answer the question, can all molecules get through the membrane? All right, this one you may have said that it can get through the membrane through the bilayer, through the phospholipid bilayer. You may have said that it can get through with a transport protein. And some of you may have even said, because you're thinking about the bubble lab, that it uses vesicles to enter. Now, can all molecules get through the membrane? Hopefully you said no, because the membrane is selectively permeable. If I were to ask you what molecules might not be able to get into a cell or out of a cell, you might say that large molecules toxins, organelles. Cell wants to keep those organelles inside of its body. Maybe inside of the cell. Enzymes, maybe enzymes need to stay in the cell and not leave. So there's lots of things. The cell is picky about what it lets in and out. So we're going to talk about how those things move in and what happens. But in order to do that, you need to understand some terms. The first term you need to understand is concentration. Concentration doesn't mean, ooh, how hard I'm thinking, let me think really hard, let me concentrate on this question. In this term, we're talking about concentration and the fact we're talking about the amount of a substance in a given area. As we increase the amount of a substance, we increase its concentration. A lot of times, concentration is measured in moles. So you might see a solution labeled as 1 m or 1 molar, and that's talking about how much of the solute is in that solution. For this class, I'm not going to make you calculate molarity. You can do that when you get to chemistry. But 1 molar would be less concentrated than 2 molar. 0.6 molar would be less concentrated than 1 molar. Sometimes we'll talk about concentration and we might say it's a percentage. So we might say that this solution is 85% sucrose. That means it's 85% sugar and 15% water. So the next term you need to understand is concentration gradient. A gradient means there's a difference in something. So a concentration gradient says that there's a difference in the concentration or the amount of something when you're comparing one area to another. So if we were to look around the classroom and look at all the students in the room and compare the number of students in the room compared to the number of students in the hall, we would say there's a higher concentration in the classroom and a lower concentration in the hall. And there's a concentration gradient when we're looking at the concentration from the classroom to the hall. So if we look at this picture, we see a concentration gradient. The one with the blue dots, we see that there's a high concentration of blue dots on the right side and a low concentration of blue dots on the left side. Now, what you need to understand about molecules is that by nature, they are in motion and they bump into each other and they spread apart. So by nature, molecules are going to move from where there is a high concentration of molecules to where there is a low concentration of molecules. So natural movement of molecules is from a high concentration to a low concentration. So if we look at this second picture, it has a picture of a cell, and inside the cell there are some red dots, some solute. And remember, solute is something that is dissolved in a solvent. So in this case, we have red solute dissolved in a solvent. Inside the cell, there are more red dots than there are outside. So natural motion, natural movement of these cells would be to spread out. Well, there's more room for them to spread out outside of the cell, so we would see a natural movement of these molecules from high concentration to low concentration, and we would see them leave the cell. Now, in contrast, in this next picture, we see inside the cell, there's a low concentration of solute, and there's a high concentration of solute on the outside. Natural motion says that these molecules are going to spread out. They're going to move from where there's a lot or a high concentration to where there is a little, a low concentration. So we would see some of that solute move into the cell. So that using that concentration gradient is how molecules move into the cell sometimes. Not all the time. There is another way that they get in, but they will naturally move from high to low as long as they can get through that membrane. So let's move on from there. But remember the terms concentration, concentration gradient, solute, solvent as we go through. So when we talk about transport, there's two major categories of cell transport. There's active transport and passive transport. And this is just to get you thinking. These two pictures are analogies or ways to help you remember things about cell transport. I want you to look at them and think, okay, in terms of concentration, high concentration, low concentration, in terms of cellular work, use these pictures and see if you can figure out characteristics of passive transport and active transport, and just write what you think about passive and active transport on these by each of these pictures. If you need more time, pause the video. If you don't need any more time, then we're going to move on. But hopefully you have some characteristics of what you think passive and active transport are. And we're going to go to the next slide, and we're going to actually talk about some characteristics of active and passive transport. So passive transport, a cell does not use energy. No ATP is needed. And when no ATP is needed, that means it's going to be easy. It's going to happen pretty quickly. It's going to happen spontaneously. And this picture, this hill, has nothing to do with how molecules move. They're not moving down or up, but they are moving from one type of concentration to another. So if we look here in passive transport, this little guy is going to go from high to low down the hill. Imagine riding your bike down a hill. Do you need a bunch of energy? Do you need to pedal a lot? No. You just kick your legs out and you let gravity do its thing and it pulls you down. Well, that's what's happening in passive transport. Molecules naturally move that way, so they're going to move from high to low, and you don't have to use any ATP. They can just move across the membrane from a high concentration to a low concentration. This is just a visual to help you remember that passive transport goes from a high concentration to a low concentration. There are three types of passive transport. There's diffusion, there's facilitated diffusion, and there's osmosis. Now, when we talk about passive transport, sometimes people will say that molecules move with the concentration gradient. And sometimes molecules move, or they move down the concentration gradient. This is just different terminology. It's all referring to passive transport. It all means that they move from high to low concentration. But think about it. If you're going with the current in a river, you're not having to use a lot of energy to paddle. You're just going with the current in your boat. If you're moving down river, you're going down the river and you don't have to paddle a lot, not a lot of energy. So this is just terminology. When they're saying it's moving with the concentration gradient or down the concentration gradient, it is passive transport, and it is moving from high concentration to low concentration. Active transport is the opposite. It does use energy. It requires ATP. And in this process, we're taking molecules and we're moving them in an unnatural way. We are making them move into a cell or out of a cell, but moving from a high concentration to a low concentration. Oh, I apologize. Sorry, guys. We're moving from a low concentration to a high concentration. I forgot what I was doing for a second. But it moves from low to high concentration. So imagine this. You're standing outside of a room, my classroom, and there are 1,000 people crammed in that classroom. And I say, you have to go in that classroom. Do you want to? No. And to get into my classroom when there's already 1,000 people, is it going to take a bunch of energy to get you pushed into that room? Absolutely. That's active transport. It's going from a low concentration to a high concentration. So here's our little guy, and he's trying to go up Transport Hill, and he's going to need a lot of energy to get there. To go from low to high, you need a lot of energy. Just like in passive transport, active transport has three types. It has protein pumps, endocytosis, and exocytosis. We'll talk about all of these different types, but a little bit more terminology. Active transport, people will say or refer to it as moving against the concentration gradient. Or moving concentration gradient. So if you think of it in terms of a river, if you're moving with the current in your boat, against the current in your boat, you're having to paddle a whole bunch to go against the current or go up the river. You're going against the natural way the river flows. So it requires lots and lots of energy. So low to high concentration, active transport. High to low concentration is passive transport. So here, if you were to look, on the left side, I have a bunch of blue dots. On the right side, I have a few blue dots. And if it was passive transport, which way would those dots move? That's right, it would move to the right from high to low. Now, same scenario. Active transport. We would have to supply energy. And when the energy is supplied for active transport, when active transport occurs, these molecules would move that way, from low to high. So passive, high to low concentration, no energy. Active transport, low to high concentration, requires energy. So keep that in mind as we start to move through these different types of transport. The first three bullet points are just reviewing things that we've already talked about, that they move from high to low concentration. It moves with or down the concentration gradient. It does not require ATP, but there are three types. There's simple diffusion, and this is where molecules move from high to low across the phospholipid bilayer. Molecules that tend to do this are small, they're nonpolar. We might say that they are lipid soluble. Now, if you look at the definition of simple diffusion, and then you look at the definition of facilitated diffusion, what's the difference? Facilitated diffusion is still moving from high to low, but it is moving through a transport protein. So facilitated diffusion is diffusion with help. Facilitate means to help. So it's diffusion with help. What is the help? The transport protein is the helper. This is where molecules, they're still moving high to low concentration. There's still no energy required, but they're going through a transport protein. This is what happens with our larger and our polar molecules. This is how glucose can move in and out of the cell. Then our third type is osmosis. Look at the definition of osmosis and compare it to the definition of facilitated diffusion and figure out what is different. Still moving from high to low, and it's moving through a transport protein. But the difference is here, we're talking about the movement of water. So facilitated diffusion and simple diffusion, this is the movement of solute. Osmosis is the movement of water. Now, for water to move through the membrane, it moves through a protein called aquaporins. And aquaporins are proteins that are specific for water. They are channel proteins specific for water. Now, there are other proteins that are channel proteins, and each channel protein is specific for the type of molecule that needs to move through it. When we're talking about osmosis, as we start talking about osmotic terms in just a few minutes, remember this little cheat, this little saying that solute sucks. Doesn't mean it's bad. It just means that it literally sucks water. So keep that in mind. We're gonna look at simple and facilitated diffusion briefly, and then we're gonna move into osmosis. All right. So simple diffusion moves through the phospholipid bilayer. And it moves from high to low. So if you notice, there's a whole bunch of little purple dots over here. There's very few over here. They are moving from a high concentration to a low concentration directly through the plasma membrane, directly through that bilayer. So that's simple diffusion. Same thing here in the second picture. Here's our high concentration. Here's our low concentration. When it moves high to low, it tells us that it is passive. It tells us that it is, there's no ATP. So it's probably going to be a type of diffusion unless it's the movement of water, then we'll call it osmosis. What type of diffusion? Simple, because it's going directly through the phospholipid bilayer. Facilitated diffusion, remember, it's facilitated diffusion with help. It looks just like simple diffusion, but instead of going through the bilayer, it's going to go through a protein. Still passive, still high to low, movement from high to low, and still no ATP is going to be required. So facilitated diffusion, the arrows show us that molecules are moving this way. Look at the squares at the top. There's a high concentration of squares. There's about 10 of them. And down at the bottom, there's one square solute. So this is our low concentration. So it is moving from high to low, but it's not going through here. It's not going through the bilayer. So instead, it is going through the transport protein.
Students have several afternoon announcements. First off, reminder that all students are still required to wear a mask, and students should be providing their own mask every day. We have extras that are for extenuating circumstances, but all students should be bringing their own mask to school. If you have an athletic block, and your mask gets sweaty, you tend to lose yours, you need to have an extra one in your backpack. The basketball jamboree is this Saturday at Benton. The girls and boys will play Bossier High School. The girls' game starts at 6. The boys' game will follow. Those tickets will be on sale tomorrow during your lunch. So if you're wanting to get a ticket, bring your money tomorrow. The cost of that ticket is $6. We will also be selling season passes for basketball, which are $75, and that will admit you to all 16 of the home games this season. Also, students, don't forget that this Friday's game team is Aloha Airlines. Students may wear Hawaiian-themed clothes and accessories. Just please remember no swim tops or tank tops, and you must wear your uniform bottoms. Thank you.
All right, guys, hope you enjoyed the afternoon announcements. So here we go. Facilitated diffusion, it did go through this protein. It's not going through the bilayer. So that's the difference when you're looking at facilitated versus simple. Simple diffusion, we would see it moving this way from high to low. But these molecules can't fit through there, so they have to use the transport protein. Going through the transport protein makes it facilitated diffusion. So we're going to look at this and see if we can identify the types of transport. First, let's identify if it's active or passive. So in this first picture, here's our high concentration. Here's our low concentration. It is moving from high to low, so that tells us that it is passive. In the next picture, high concentration at the top, low concentration at the bottom. We're looking at the concentration of blue dots. So it is moving from high to low, so that tells us that it is passive. Third picture, there's a low concentration at the top. There's only 3. And down at the bottom, there are several. So this will be our high concentration of blue dots. So it's moving from low to high going on transport hill. If you looked at the picture, they'd be going up the hill from low to high. So this is active, and it would require ATP. So now let's look at these first 2 drawings again and see if we can say what type of passive transport it is. These are moving directly through the phospholipid bilayer, so that means it is, that's right, simple diffusion. And in the second picture, it's moving high to low, it's passive transport, there's no ATP required, but instead of going through the bilayer, it is going through a channel protein. This is our helper. And remember what word means to help? Facilitate. So this is facilitated diffusion. Now, osmosis, when we talk about osmosis, it gets a little bit more complicated. We add some terms that you have to know. So there are 3 different terms. It has to do with tonicity, how much water is in a solution. So something that is hypotonic, the prefix hypo means above or more. So a solution that is hypo, I apologize, scratch that. Hypotonic means below or less. I apologize. Below or less. So a hypotonic solution has less solute. Hypertonic, hyper means above or more. So a solution that is hypertonic has more solute. An easy way to remember that, what kids are going to get more hyper? The ones with lots of sugar, a solute, or ones with very little sugar? Right. The ones with more sugar get hyper. So hypertonic, more solute. And then we have isotonic. Something that is isotonic, iso means equal. So isotonic has equal amounts of solute. Now, cells react differently to different solutions. If we put a cell in a hypotonic solution, so here's a picture, here's our cell, this outside has less solute, so there's lots of solute in the cell, very little outside. That's hypotonic. Remember that cheat I told you, that solute sucks? So wherever the most solute is, it's going to suck the water in. So there's more solute inside the cell. What's outside the cell is hypotonic. So this solute sucks the water in, and the cell swells. A way to remember it, hypo-hippo. It's going to swell like a hippo if you put it in a hypotonic solution. If we put a cell in a hypertonic solution, there's some solute in the cell, but there's lots of solute outside of the cell. Remember, solute sucks. So wherever the most solute is, it sucks the water out. So it's going to pull the water out of the cell, and the cell is going to shrivel. It's going to shrink. In animal cells, we say it crenates. In plant cells, we say it plasmolyzes. Those are just fancy ways of saying it shrinks. So this isn't an animal cell. It crenates. In plant cells, it plasmolyzes. In an isotonic solution, we have a cell. But this amount of solute is equal inside and out of the cell. So what happens is what we call dynamic equilibrium. Some molecules move in, some water moves in, some water moves out. But the cell stays the same. It's dynamic equilibrium because they are at equilibrium, but there's still water moving back and forth. Now remember, whenever we are talking about this, we are talking about the movement of water. The solute is not moving. The membrane is not permeable to whatever solute we're talking about. So we're talking about the movement of water back and forth. So if we look at these pictures, we have a red blood cell, and we put the red blood cell in a solution, and we can see that water, there's lots and lots of water moving in. So it's swelling, it's getting bigger. We would say that this solution right here is hypotonic. This solution right here is hypotonic because it's swelling like a hippo. Now, if this animal cell, it doesn't have a plant cell wall, so if this animal cell were to take in more and more and more water, what's going to happen to it? Yep, it's going to explode. But we will say that the cell will lyse or split, explode. Then we look at this picture, and we can see that water is moving out. There's some moving in, but the majority of the water is moving out. So the cell is shriveling up. That's what we said, it was crenating. Well, when the cell crenates, the cell shrinks, it's losing water. That tells us that the solution that it is in a hypertonic solution. Then we look here, we see a happy, healthy red blood cell. We have an equal amount of water moving in and an equal amount of water moving out. So this is an isotonic solution. So let's see if we can identify what type of cell each solution is in. So this cell shrunk. If it shrunk, what happened? It lost water. If it lost water, what type of solution would it have been placed in? Remember, solute sucks. So that means there's more solute out here. If there's more solute out here, that means it is a hypertonic solution. Now we look, here's a nice, normal, healthy red blood cell. It's not getting bigger, it's not getting smaller, so that means if water moves in, the same amount of water is moving out. It is pretty equal, so it is isotonic. Then here we have a cell that is swollen. That means it has taken in a ton of water. If it's taken in a ton of water, it means that there's more solute in the cell. There's very little outside of the cell. So outside of the cell is hypotonic. I hope this is starting to make sense to you. So let's think about our egg. In our egg experiment, on day 1 it looked like a regular chicken egg. Day 2 the shell was off and the mass increased. So if mass increased, that means that vinegar moved into the cell. So what type of solution was vinegar? If water moved in, it meant there was more solute inside than there was outside. So it means there's very little solute outside. So it is hypotonic.
Remember to stay 6 feet apart and wear your mask correctly.
In terms of
Walk directly
osmosis,
to your designated area.
what type of solution was the corn syrup? Well, in the corn syrup, you can see it shriveled up under the day 3 picture, and we saw the mass decrease. So since the mass decreased, it lost water, which meant there was more solute outside of the cell, and it sucked the water out. Corn syrup is very thick, lots and lots of sugar, so it pulled the water out. So it meant the corn syrup was hypertonic. And then on day 4, we put it in distilled water, and we put it in distilled water, water moved in. The cell got bigger. It got really swollen. The mass increased, which tells us that there was more solute inside of the cell, inside of the egg. So the solution outside had very little solute. Remember, it was distilled water. It was pure water, so it had nothing but water in it. So it was hypotonic. All right, so we're going to practice just a few more. And if we look at this picture here, it tells us that the red molecules are sugar, the blue molecules are water. And this is what it looked like when we started. Lots of solute. All right, guys, sorry about that. All right, so we have a high amount of solute because remember, sugar is solute. So this side has high, this side has low solute. We have to decide which way is the water going to move. So then we look at this picture, and if you look, there's 3 molecules of sugar on this side. If you look at the ones on the right, there's still 3 molecules of sugar on the right side. So the amount of sugar on each side didn't change, but what happened, look at your level of water. Water moved that way. So if water is moving, what type of transport is it? That's right, it's osmosis. And if it's osmosis, how would you classify the solution on the left? It sucked the water in, so it was hypertonic because it had more solute. Now, you could also say, well, what is the solution on the right? It has less solute. If it has less solute, we could say that this solution is hypotonic, meaning it has less solute. All right, so we're going to say what type of solution is in each of these beakers and which way will the water move. So here we have a solution, and inside the cell, it's 10% salt. I don't know how easily you can read that. And in the solution in the beaker is 10% salt. Salt is a solute, so our solute equals each other. And since our solute equals each other, we would have equal movement of water in and out, and this would be isotonic. Here we have 2% salt, and here we have 10% salt. So inside the cell has more solute, outside of the cell has less solute. So which way does the water move? Remember, solute sucks, so water moves into the cell. Less solute tells us that the solution that the cell is in is hypotonic. Then in the last beaker, we have 30% salt, and inside the cell we have 10% salt. So there's more solute outside of the cell. Solute sucks, so wherever the most solute is, that's the way the water will go. So water moves from the cell into the solution, so the solution outside is hypertonic. Now you might be asking the question, I thought passive transport was movement from high to low. And it is, because we were talking about the movement of water, right? But to figure out where the water is moving, it's easy to look at the solute. But let's look at it. We said water is staying the same. So if 10% is solute, we know 90% in the beaker is water. If 10% is solute in the cell, we know 90% is water in the cell. So there's equal amount of water as well. Well, here in the cell, there's 10% solute, so there's 90% water. Outside of the cell, there's 2% solute, so there's 98% water. Which way did we say water was moving? We said it was moving into the cell because there was more solute. But if you look, water is actually moving from 98 to 90. It's moving from high to low concentration. Then if we look here, if we have 30% solute, it means we have 70% water. And if we have 10% solute in the cell, that means we have 90% water. And we said the water is moving out of the cell. It's moving to the most solute because solute sucks. But if you look, water is moving from 90% to 70%. Water is actually moving from high to low. So osmosis meets the requirements for passive transport. It's just very easy for us to look at it and go, wherever the most solute is, that's where the water will go. All right, so let's look at these pictures. What happened in these pictures? We have celery, and we have one celery that's standing up straight and tall, and we have another stalk of celery where it's all bent and limp and wilted. So the one that's wilted, what do you think it has lost? Why is it wilted? Yep, it lost water. Well, if it lost water, what could you tell me about the solution it's in? If it pulled the water out of it, it would have to be have more solute, because solute sucks the water out, so it is hypertonic. Now this celery is standing up nice and straight and tall. It hasn't lost any water. What's going to make the central vacuole fill up with water? Water going into the cells is what's going to make those vacuoles fill up. So if water is going to go in, that means there's less solute in the solution that it's in the water, so less solute. So it tells us that this water the celery is sitting in is in hypotonic, is a hypotonic solution. Active transport. In active transport, remember it's the opposite of passive. Molecules move from low concentration to high concentration. They go against the gradient, up the concentration gradient. It does require ATP. Now there's three types. There's ion pump, proton pump, and protein pump. There's also endocytosis and exocytosis. We're going to talk about each of these briefly. You just have to have a basic understanding, but the biggest thing to remember here is that active transport moves from low to high and it requires ATP. Biggest thing to remember about active transport. The ion pump and the protein pump, or proton pump, protein pump, what it's doing is pumping in molecules that are charged, they're too big, but it's pumping them against the concentration gradient. So if you look in this picture, here's our high concentration of these hydrogen ions. Here's our low concentration. There's a bunch in there, but we're going to keep shoving them in. Well, to shove them in, we have to use ATP. We have to use energy. That it's literally pumping it in. If you see the word pump, you need to assume it's active. Next type of active transport, well, this is just going through it again. It's giving you a definition of it. It is moving it from low to high. It's forcing those molecules in. Then endocytosis. In endocytosis, a cell engulfs large molecules and it takes it in. It does take in it requires energy to swallow it. This one is hard to look at and say, is it moving from low to high? Technically, it still fits the requirements, but what you need to look at and say, is it going to take a bunch of energy for a cell to literally come and wrap its membrane around it and take it in? This is what we see when an amoeba is eating. It goes and it literally wraps itself around particles. See how the membrane dents in? The food enters, it wraps its membrane around it, and then it leaves in a vesicle. So all of these pictures are showing the same thing. The membrane indents in, particles are surrounded by a membrane, and then a vesicle goes into the cell, and it takes this large amount of food in at one time. Now there's two different types. There's cell drinking and there's cell eating. Pinocytosis, cell drinking. So you can think of pine salt or you can think of pina colada. It's liquid, cell drinking. Phagocytosis, phago, food. Phago, food. Phagocytosis, it's taking in food. So if you look, here's just showing you pictures of endocytosis and phagocytosis and pinocytosis. Here you can see it's taking in large amounts of something. These blue molecules here are going to be some liquid form. It can be like a cytoplasm, a gel-like form too, but it's considered pinocytosis. But here we have a large particle food that is being taken into the cell. And remember, the cell is engulfing it and just taking it in. Then we have exocytosis. Exocytosis is the exact opposite of endocytosis. Literally, something is leaving the cell. What type of things might leave the cell? Proteins, hormones, waste. And what happens is a vesicle containing whatever needs to leave the cell begins to fuse with the membrane. And as it fuses, it opens up, and then once that vesicle opens up, those molecules are excreted from the cell. And that's exocytosis. It does take energy for this to happen. Remember, a vesicle is made out of a lipid bilayer. That lipid bilayer matches the membrane. So as a vesicle fuses, it's all the same material, and it just fuses, and that vesicle becomes part of the membrane, and the materials are excreted. And this is just showing you other examples of exocytosis so that you can see these molecules leaving. And remember, it's waste, hormones, enzymes, chemical signals. Any of those things can be excreted from the cell. So let's see if we can identify what type of transport each of these is. First, let's identify high to low or low to high. So for this picture, we're going to divide it out. Let's look at each picture individually. It's going from high to low concentration, and it's going through the phospholipid bilayer. So we would say, because it's from high to low, it's passive. And because it's going through the lipid bilayer, it is simple diffusion. Looking at the next picture, looking at our orange solute, it is, our high concentration is here, our low concentration is here. It is moving from high to low, so that tells us that it is passive. But it is going through a transport protein, so it has help. So it is facilitated. And then in this last one, we have 2 molecules here, we have a bunch down here, so this is our low concentration, and this is our high concentration. So it is moving from low to high. That tells us it's active. If we were a little confused, we see ATP, which tells us even more so that it's active. If you see ATP, it's going to be active. But here's the deal. They don't have to show ATP for you to know it's active. If it's moving from low to high concentration, we know it's active. Now, this one, we see a protein and it's going through a protein, so we could say that it is an ion pump. You could call it a protein pump. Or even a proton pump, meaning it's moving positively charged molecules through. All of those are correct, but a protein pump is fine. All right, let's look at another picture and see. We're going to see if we can identify what is happening. So in this picture, at the top, see the arrows, we see them coming in, and it's being engulfed by a vesicle, so we would say it is endocytosis, and it is active. Here, we see a vesicle fusing with the membrane and releasing something. We would say this is exocytosis, and it is active. In this picture, we see a large food particle coming in. Follow the arrows. It is a solid particle, so it is endocytosis. It is active transport, and because it is a large particle, we could also say that it is phagocytosis. Now here, shows a molecule leaving. These molecules are being expelled, so it would be exocytosis, and it would be active. In this picture, we see vesicles, and they are traveling to the membrane, and they are releasing molecules. These are secretory vesicles, which means they're going to secrete something, that they are releasing something out. So this would be exocytosis, and it would be active transport. Arrows are important. Make sure you look at arrows when you're looking at diagrams. Then we have this picture. Low concentration of green squares, high concentration. Also uses ATP. The arrow says it's moving this way, so we would say it is active. And protein pump. Then we see this picture. We have a membrane, and it shows that there is movement this way. There are no sucrose molecules on this side, so it tells us that sucrose molecules are not moving this way, and it tells us it's differentially permeable, meaning selectively permeable. So what is moving? Is the sucrose moving, or is the water moving? The water is moving. So what process is this? It is osmosis. And what would we call this solution on the right with all of the sucrose? It's sucking the water in. So we would say it is hypertonic. And the solution on the left, it's losing water. So if we had to compare it to that one, we would say it has less solute. So we could say it is hypotonic. In this picture, we see molecules going from high to low. In both circumstances, they're moving from high to low, and they are going through a protein. So high to low tells us that it is passive, and through the protein tells us that it is facilitated diffusion. All right, so we look at this picture, and we have our solute molecules at the top and the bottom. There are more at the top, so this is our high concentration, less at the bottom. So this is our low. How are they moving through? You can see them. They are moving through the phospholipid bilayer. So because it's moving from high to low, we know that it is passive. And because it's going through the bilayer, we know that it is simple diffusion. All right, we're going to look, we want to identify whether the solution is hypertonic or hypotonic. So if we look at this, here's our membrane. On this side, we have 0.4 molar glucose and 0.5 molar salt. So how much solute do we have total? We have 0.9 molar solute. And on this side, we have 0.8 molar glucose and 0.8 molar salt. Glucose is sugar, salt, both of them dissolve in water, so they're both solutes. 0.8 and 0.4 give us 1.2 molar solute. So which side has more solute? The one on the right, so it is hypertonic. The one on the left has less solute, so it is hypotonic. Which way will the water move? That's right, hypertonic, more solute. We know solute sucks. So we know that it will move this way. Now this tells us that the membrane is not permeable to either solute, so we know that this is water moving. So we know that this is osmosis. So now we want to identify the solution outside of the cell. Inside the cell is 0.2 molar glucose. Outside the cell is 0.2 molar glucose. So we know that this solution is isotonic. That should be a C. Isotonic because the solute in both are equal. So we're going to look at these. We want to identify the solutions, the actual solution the cell is in. So what is in the beaker? What is the cell sitting in? So we're going to look at it. Inside, there's 0.25 or 25% sugar. Outside, there's 0% sugar. So there's less solute outside. So the solution outside the cell is hypotonic and the water would move in. Here, there's 5% sugar outside, 25% sugar inside of the cell. So there's less solute here, so this solution would be hypo and water would move in. Then we get here, and there's 10% sugar on the outside, 25% sugar on the inside of the cell. So less solute outside, still hypo, and water moves in. Then we get here, and we're moving along. 15% sugar on the outside, 25% sugar on the inside of the cell, still less solute, so it's still hypo, and water moves in. Here, 20% sugar on the outside, 25% sugar on the inside of the cell, so there's still less solute in the beaker, so it is still hypo, and water will move in. Then we get to this picture, there's 25% sugar outside and 25% sugar inside of the cell. They are equal, so we would say that the solution is isotonic. Now, here's my next question. Which one of these cells would gain the most mass? That's right, this one. Because there is such a difference in the solute, more and more water is going to move in because it's trying to even it out. Which one is going to gain the least mass? Well, obviously this one. The mass would stay the same. All right, guys, so here's what you need to know. Tomorrow, you need to study for your 15 and 16 prefix suffix quiz. Tomorrow, we'll look at this flow chart. You'll get a chance to fill it in. We'll do some activities to have you apply what you just learned about osmosis and diffusion. So I hope you're understanding it. If you're not understanding it, send me an email, ask me questions in class, but I will see you tomorrow. Y'all have a great day.